Okay, cool. So the thing that I was about to say is, this is the first time I've done an undergraduate course on Zoom. I've done a couple of lectures and somebody else just said, here's the thing, you go. So there's going to be some glitches, like I forgot to record. So please help us out. And if something weird happens, don't be bashful about letting me or the TA know that, no, dude, you got to stop and start doing something else. Okay, so that's a disclaimer from the very start. I'm going to screw up. Okay, so help me out. Right now, the enrollment is set at 50. I tried to get it up to 70, but it was curtailment and the registrar was closed. So hopefully we're going to increase the enrollment cap to 70. I think there was 29 on the wait list. Generally, 9-ish or so will drop by then. Anyway, if it turns out that there's one person on the wait list, I'll do a PTA so you can still take the class. Okay, so you're probably in. But if you're not going to stay in, just go ahead and, if you hate me, which has happened, go ahead and drop it and take something else or whatever. But hopefully we can get everybody in. That's the ultimate objective. Okay? Okay, so I'm going to go and share screen and show you a bunch of slides. While I'm doing that, I can't see you guys very much. I can see maybe one or two of you, and I can't see the chat room, and I can't see the hand raised and all that stuff. So I'm going to ask the TA, Corey, to monitor that. And if you have a question, raise your hand. And then when I stop and say, are there any questions, or if it's urgent, Corey can go ahead and interrupt me and we can do it that way. Okay. Otherwise it can be chaos, as you know.
Hey, Greg, this is Leah. Can I introduce myself?
I'm going to introduce you in a moment.
Okay.
Okay. Okay. Jumping the gun there.
Okay. We actually do have one question real quick, and it's just pertaining to the Zoom link. So for each class session, will they be able to use the same Zoom link that was published?
I don't know. Okay. So I set it up every Monday, and then I set it up every Wednesday, and those Zoom links are different, but I think they're all my Zoom links. So I think it should be okay, but I don't know for sure. I guess we'll find out Monday is the answer, right? So Corey, why don't you try the Monday link on Wednesday, and we'll see if we see you. And at 9:01, if you're all by yourself, then we'll know the answer.
All right, sounds like a plan.
Okay, I've got an experiment going already. Okay, share screen. This one. Okay. Okay, so the instructors are myself. I just introduced myself. And the other is Leah Krubitzer. So Leah, can you go ahead and introduce yourself now?
Okay, yeah, hi. Hi guys, my name is Leah Krubitzer. I'm gonna sort of co-instruct this with Greg. Normally we have a really large lab component of this class, but because of COVID, we can't do that. So we've sort of modified a lot of the lectures so you can learn a little more about neuroanatomy than you normally do. So I look forward to teaching you guys in the next few weeks. Hey.
Hi, thank you. And Corey, why don't you introduce yourself? Corey is our Crackerjack TA for this class.
Sure. Hey, everyone. My name is Corey. I'm a first-year graduate student in the biopsych program, and I'm going to be your TA for the quarter. And so when it comes to having questions about the course and going to midterm reviews right before you take your midterms, I will be handling all that. So please, if you need any more resources or need any help with anything, please contact me or go to my office hours.
Okay, thank you. Okay, so as we already touched on, lectures are Mondays and Wednesdays. I like to start right on at 9 o'clock, and I hate to go all the way to the end. So I usually quit early. Okay, so everything's in the beginning. It's the Zoom invite. Hopefully this won't be an issue tomorrow. I really kind of want you guys to come to class if you can, right? I realize some of you might be in a time zone where it's 2 o'clock in the morning right now or something like that, and that's okay. We're making it asynchronous to help to accommodate everybody who wanted to take the class. But I'm going to hopefully do some chat rooms and do some other innovative teaching kinds of things that I don't think work well during the recording lectures. And you're, of course, always welcome to relook at the lectures afterwards, right? So I'm going to post them as soon as I can after class, okay, which will be around 11 o'clock probably, as I'll tell you in a second. There's a couple of resources that you can use, the comparative mammalian brain collections. I'm going to show you some slides from that today. and the Allen Brain Institute Atlas, which we'll talk about more tomorrow. And then usually, at least for my lectures, there's lecture notes that are included. And I really want you to read those too, because that's fair game for the testing material. Okay, so read that. It's 5-7 pages, something like that. And I deviate in the lecture from the lecture notes, so it's not just me reading the lecture notes or anything like that, but it helps put things into some context. If you read it before the class, actually, I'll make a lot more sense. Okay. Any questions so far? So far, so good? Okay. So office hours, Leah's are Monday right after class. Mine are Monday and Wednesday after class. And Corey's are shown here on Tuesday, Thursday, 5 to 6. Okay, and there's a contact information you can get. I'm just pulling this right off the syllabus. This is posted on Canvas, so you already have access to all this. I just wanted to go over the lecture schedule a little bit to give you some ideas of what we're going to be doing. So here we are at lecture 1 right now with me in blue and Dr. Krubitzer in green. And the first chunk of the class, the first 5 lectures, is going to be about how you do neuroanatomical experiments. What does comparative neuroanatomy mean? What are the different techniques we can use? What it is that you can and can't answer using these different techniques? And what does the data kind of look like? Right? And then we're going to have the midterm. First midterm 1, January 25th. It's 100 points. And so what to expect? So you're on your own. Okay, everybody does their own test, right? The questions are going to be largely factually based, which means there is an answer that you can find. Okay. Sometimes it'll take more than, the answer won't be on a slide. You'll have to think about a couple of different concepts or ideas and put them together to come up with the answer, but it's not going to be, super imaginative in that sense. You have 24 hours to do it. We wrote it so that it should take you, if you're well prepared in a classroom setting, about an hour, right? And if you want to spend the extra 20 minutes for the hour and 20 minutes you would normally get to do this, I know some people like to do that and go over your answers and change them back and forth a few times, you would have plenty of time to do that. So we're giving you 24 hours. Again, so if you're in some strange time zone, not strange, some distant time zone, and it's 2 o'clock in the morning now, you'll have from 2 a.m. to 2 a.m., right, to get us the answer. Okay? This is stuff that you just got to know. Okay, I don't like to use the word memorize, but you just have to know what the different words mean. Okay, and you have to know it. So this is just an exercise really for you to show everyone, yourself included, that you do know these terms, you understand these basic concepts about directionality and things like this, so that when we're lecturing beyond this, you can keep up. Okay, so this is just kind of, the reality of science. You got to understand what the words mean. Okay, so after that, then Dr. Krubitzer will give you one more lecture to explain, kind of why we're using these approaches. And then you're going to get a big dose of me on examples of receptors and the auditory system and the visual system. And then we're going to get a holiday in there. And then comes midterms 2 and 3. You'll notice that midterm 2 is on Monday and 3 is on Wednesday, 2 days later, which sounds pretty stupid, right? But that's because it's really one big midterm in 2 parts. So what to expect for those? So I'm going to assign you into groups of students, 4 or 5. It depends on how many end up being enrolled in the class by then. Each group is going to be given a question, and it will take 24 hours to complete it, and that's for 100 points. Okay, so I want you guys to work together, and the question isn't going to be something that you can see from the slides. It's going to be something like design an experiment, okay, something like that. And so you're going to have to think about it, right? Normally, you could do this in about an hour, hour and a half, right? So you'll have plenty of time to do that and really think it through. And I really don't want you to spend 24 hours on it because you'll just drive yourself crazy, okay? But make a good effort at it. Come up with your answer, finish it off, turn it in, take a deep breath, have fun on Tuesday. And then on Wednesday, the midterm 3 is your same group, your same 3 or 4 colleagues are going to grade 2 or 3 other groups as answers. Okay, so this way, and they're going to be different questions, right? So it might be the same question you got, might be a different question. Okay, and I want you guys to think about, how their answer fit in with what the assignment was. So we'll give you the rubric and you'll know what you're supposed to do so that when you're doing midterm 2, you know what the answer, and when you're grading, which is midterm 3, you'll know what they were supposed to do. This is supposed to help you learn about how other people think and how you can get this all to work. Okay. Then what we're going to do is look at your grades for midterm 3 and see how close are they to what you should have said, for lack of a better word, and why. So when you're grading, you can't just say, I didn't like it that much. I'm giving it a 70 out of 100. Right? You have to explain, no, 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 you did this wrong, you did this right, you did this okay, et cetera, et cetera. And therefore I took off 5 points for this and I took off 2 for that. Exactly what you want to see when you get your grade back, right? You don't want to see a grade that says 70, so-so, right? Because that's no feedback. You're not going to know how to do better. So the idea is for you guys to tell your classmates how to do better, and then for us to tell you guys how close you were at doing that, okay? Are there any questions so far? Okay, this is the first time we've done it this way, and I expect to need to modify it a little bit. So, you know, we're kind of experimenting on you in a sense of how this is all going to fly. Are we good, Corey?
Yep, we're good.
Okay, thank you. Okay, and then we have 4 more lectures. Talk about the somatosensory motor system a little bit, and then the last 2 lectures are going to be a version of what Dr. Krubitzer and on the 8th and me on the 10th, what we give to our colleagues when we give a scientific talk. Okay, so normally we'll talk to a bunch of other neuroscientists and graduate students, postdocs, professors, et cetera, and tell them about our work. And so at this point, this is, you're going to be able to understand a great deal of that because you've already taken this course. And then it'll give you a flavor of, you know, what's it really like to sit in through one of these kinds of courses, which most of you probably haven't yet, right? But you're certainly capable and allowed to do that, right? So this is hopefully going to give you the confidence to go, you know what, I can sit in and listen to some Nobel laureate, not me or Dr. Krubitzer, but they come to Davis sometimes, and I can listen to them and understand what they're saying. Okay. And then we're going to have the final exam. Oh, did you see my dog? Did you see that? No?
No, I don't think so.
Okay, fair enough. Okay. Usually I get an awe when they see my dog. Okay, final, what to expect. So the final is optional. Okay, if you got a good score on your 3 midterms, right? You got 100 on the first one, which you're probably going to do, and you got A-ish values for your midterms 2 and 3, and you're, you know, like, well, I'm fine with that, then you don't have to take it. Okay, we'll just take your 3 grades. If, on the other hand, you're like, man, I totally screwed up. My group is a bunch of idiots. We didn't know what the hell we were doing. I got to take the final because we hardly got any points on midterm 3 because we didn't know what we were doing, or because that one person in my group did something stupid or whatever, right? I think I can do better. Okay, in this case, you get to pick your own group, even if it's just you, okay? And you get up to 4 colleagues, so you can have a group up to 5 again, right? Or you can have 2 or 3 or 4 or 1, right? You only get 2 hours to complete the exam. It's going to be a grant proposal, and you're going to have a background and a significance and methods and expectations and conclusions, et cetera. Okay. Well, I've done this before, and I'll give you examples of how that's done. And you can do this in about 2 hours, right? You end up with 1 page, and then you turn it in, and that will take the place of your worst midterm of 2 and 3. Everybody should get 100 on 1, so that's not going to even count. If you only get 50 on 1, then you should probably drop the class. Okay. Does that make sense? Okay, so it's entirely up to you. Right? If you do have colleagues, if there's 5 of you do it, all 5 of you get the same grade. Okay, and you have to tell me beforehand that that's what you're going to do. Okay, you have to tell me before Friday. So I'll know what to expect. You can't go, well, you know, I decided after all, oh, I decided to take it and it was harder than I thought, so never mind. Right? No, let's just go for it. Decide and do it. Okay? Okay. Okay, now I need some feedback. So I have a few undergraduates in my lab, and I've asked them a couple of times, what's the best way to do a Zoom course? And I've gotten a lot of feedback that says Yellow Dig is awesome. You should really do Yellow Dig. I had never heard of Yellow Dig before that. I looked at it over the weekend. I saw their propaganda film or whatever, and I'm a little bit on the fence on it. So I'm asking you guys, do you like Yellow Dig? Is it a fun thing to do? Is it totally stupid? let me know. Okay, so go ahead and chat and et cetera, and just give me some insights into whether I should even bother pursuing this. What I'm thinking about doing is, since I've already have the points set up, is that historically what I've done is start right at 9 o'clock. And usually if there's supposed to be 50 people in the class, there's maybe 20, right? Especially after the first midterm. And I will say something like, my dog's name is Rico. And then on the exam, there'll be a multiple choice question that'll be a bonus point. And it is, Dr. Rickenzone's dog is named Fido, Rico, Spot, or Fluffy, right? And if you were in class, you'd know it was Rico, and therefore you got a so-called bonus point. Okay, and the way the bonus points work is I take all your points, except for the bonus points at the end, curve the thing and say, okay, well, to get an A, you needed 250 points or whatever it is. And so, oh, you only have 248, but over the course of the quarter, you managed to get 5 bonus points. Therefore, now you have more than the cutoff, and so your B plus becomes an A. Okay, so that's what bonus points are about. And I'm thinking that's probably what we'll do here, right? So if you participate and you do the yellow dig thing, you'll get some number of bonus points, probably on the order of 10 or something like that, but enough to scoot you over across the line and turn your B plus into an A minus or A minus to an A or whatever it is, okay? As opposed to actually forcing you to do this. So if you think it's stupid and you don't want to bother because it sucks, you don't have to, right? But if you kind of like it and you're getting into it, then you get, you know, a bonus for that. Okay, does that make some kind of sense to everybody? Corey, does anybody have anything to say about this?
Just to inform, not many of the people here have ever used Yellow Dig. I did find the website and I am publishing it in the chat now.
Super.
Is like another platform similar to Canvas or to Zoom where you give out assignments?
It's similar to like Piazza. In my experience, I used it in a NPB course last quarter, and I liked it. It was nice to kind of connect with people, but it just kind of depends on how you go about it, I think.
Anybody else have a positive experience with it? Anybody have a negative one?
We have one student that's still confused about how Yellow Dig is going to be part of the bonus points.
Yeah, so so am I. Okay, so one student and I think alike. So I watched the film, and it says you can get these certain number of points, and it would probably be like 1 point a week is what I'm thinking at this juncture. And you have to go on there and you have to post something or hang out with or make a comment or whatever. And then if you do enough of that, then you get a certain number of points. And how exactly I would implement it, I don't know, right? So I just didn't want to get too invested in this if people hated it. I don't know, does it cost money for you guys? Because it's free to me, but I don't want you guys to have to spend.
It was free, I think. So it should be good to go as far as free-wise.
Okay, good. I don't want you guys spending money on this.
I will say that if it's anything similar to Piazza, I would recommend it because I used Piazza back when I was trying to code for MATLAB, and having it all available there, the response times of the TAs is very helpful, and it's kind of more of a problem board, but a troubleshooting board. If any of you have used forums before, it's kind of like that.
Yeah, that's my understanding. It's like a forum. Okay, well, if you feel strongly about it one way or the other, you can always email me, and then I will probably go ahead and see what I can do. Okay, and if it's an unmitigated disaster, then I'll deal with that too. Okay, I'll sue them and give us all a bunch of money or something. Buy pizza from Grubhub or something. Okay. Okay, this is also in the syllabus. This is important because this is weird, right? I mean, we're in this epidemic. We're taking classes in our pajamas. I'm not wearing any pants, that kind of thing, right? So if you need support in any way, even if you think you might, don't hesitate. Go ahead. You have lots of resources that you can go ahead and take advantage of, and I encourage you to do so. Okay, so just know that stuff is handy, and it's on the Canvas site So you can go straight to the horse's mouth here and see what to, how you can get some help. Okay, if there's no further questions, I'm going to do a little bit of lecturing now. We good, Corey?
Yep, you're good to go.
Okay, good. So
I have just one question.
sure.
Do you recommend any programs or virtual lab apps we can use off of Steam or anything online?
I don't, Leah. Do you have any suggestions? Are you still
There's a
here?
couple of virtual labs online, if like through Steam, if you have a VR set, like an Oculus or something like that. I do know there's a couple of rat dissection virtual labs online due to previous biology courses.
If you have access to a VR set, I also have access to Steam. I can look into that for you. If that would be helpful for you, I can totally do that. And to anyone else who has the ability to have a VR set and or just has Steam capabilities, I can look into it.
I'll send the wish list with the stuff on it.
Okay.
Great. Thanks, Corey.
Yeah, so I'm here. I mean, my plan wasn't to do anything like that, just to give some gross brain anatomy. And I have some videos of our rat surgeries and how you use a stereotaxic atlas and go about doing those things. Because normally what happens is we actually do in the lab live rat surgeries. The students do them hands-on. We teach you how to do all these surgical techniques, but it's just not possible. So it's going to be a lot more limited this time.
Well, does buying freeze-dried rats at the pet store work? I mean,
No,
technically they're already sacrificed.
no, that's not. Greg, are you going to talk about doing research and IACUC approval and all the kind of stuff? You can't go buy rats from the pet store and do experiments on them.
Yeah, I'm about to
freeze-dried rats.
What's that?
Freeze-dried rats, like the type you feed.
Live rats. I'm not certain how helpful it's going to be because, first of all, if the rats have been frozen, my guess is the tissue in the brain is just going to be crap. And so I'm not quite certain how much you would learn under those circumstances. Rat brains are pretty small to start with. I mean, if you want to do that as a civilian, I can't stop you from doing that, but I'm not certain in terms of neuroanatomy, you know, taking out a frozen rat brain, it's probably going to end up being mush. And like I said, it's really unfortunate for all of us that this is Zoom because the lab component of this course is something that the students really, really love. It's about 7 hours a week or more that you have to put into the lab. And we teach you how to do an experiment on a live rat, recover the rat. So monitor the rat, do the surgery, do the brain surgery, inject anatomical tracers, euthanize the rat, perfuse the rat, cut the brain tissue, stain the brain tissue, learn how to do a microscopic analysis. So it's just, unfortunately, you guys aren't going to get to have that experience. But, you know, I think, you know, doing a dissection on a freeze-dried rat, you're not going to really get much out of that. It could be cool as for you if you want to go and do that. But in terms of brains, what I did over the holidays, I'll talk about this on Wednesday, with my mother visiting, I got a whole bunch of sheep brains, which I dissected, cut in different planes. I'm marking them. So I think that's going to be useful for just gross brain anatomy. I'm hoping throughout I'll show you some microscopic comparative slides of different animals as well. So I tried to pull together the best I could. It's not going to be brilliant, but I think I think it's going to be pretty good. I hope. So anyway, Garrick, yeah.
Okay. Okay, well, that was actually a really good intro because what I'm going to talk about now for the next 20 minutes or so is what it takes actually to do experiments in vertebrates in a place like UC Davis. Okay, so there's a number of animal issues and it's the care and use of laboratory animals. And I'm going to go over kind of what the rules are. Back in the 70s and early 80s, you could do whatever you pretty much wanted to. You go to the pet store, buy a bunch of rats and, you know, do experiments on them. And nobody oversaw that, right? But nowadays there's quite a bit of oversight. And I just want you all to have this common understanding that it's not the simple case that if you have a scientist with money has an idea in their head, they can go out and do a bunch of experiments on a bunch of Shetland ponies or something, okay? So you have to understand what that is. Okay. Okay, so who regulates animal care and use? There's the U.S. Department of Agriculture, the NIH, this organization called ALAC, and it's all combined here with the so-called IACUC. So what do all these words mean? So USDA is U.S. Department of Agriculture, so those are the feds. NIH and the Public Health Service, whoops. That's the National Institutes of Health. Right? These guys are important because they give scientists like Dr. Krubitzer and myself the money that we need to pay the people and for the animals to do the experiments. Okay, so they hold the purse strings. There's this acronym called ALAC, which is the Association for Assessment and Accreditation of Laboratory Animal Care International. This is a group of folks that come around every 3 years to every university that has NIH money and make sure that we're following all the rules, okay, as I'm going to be describing them. So this is an independent body that makes sure that everything is fine. And then there's the Institutional Animal Care and Use Committee, which we're going to talk about quite a bit. And that's the in-house committee that makes sure that every investigator follows all the rules. Okay, so the feds had mandated a number of different policies that one has to follow. And essentially they said all institutions have to self-regulate via this IACUC. Okay, so if the IACUC, if you ask IACUC, can I do these kinds of experiments? And they say yes, then it's okay for you to do them. If they say yes and it's wrong, in the sense that it's breaking laws, they're the ones that get in trouble, not you, because you got permission to do it. And so the oversight stays with the so-called IACUC. And so what they do is they approve the proposed animal use before any animals are obtained or the work is begun. So what you can't do is say, I got this great idea that I think is going to solve, world hunger, and I'm going to pilot it in a handful of rats, and to make sure it works, right? And then I do that and it works, and then I go, oh, can I do these experiments? And ask IACUC. You can't do that. Before you use any animals, you have to ask IACUC, I have this great idea, it's going to cure world hunger. I want to use the rats this way, may I please, right? So you can't say, I know it works because I already did it. There's semi-annual inspections. They inspect all the animal facilities, so all the vivariums where the animals are being kept, the lab and study areas, and then they make sure that these protocols, which we'll talk about in a second, are, you're still following them, doing what you said. Okay, and if they find out that you're doing something wrong, then they have to report it to the various agencies, commonly the USDA. Okay, so it's required for any live vertebrate animal work in teaching or research. I don't know why live is in quotations, but one reason is, as we just discussed, you can go and buy a dead frozen rat from Petco or wherever and do experiments on that. Okay, even though a chicken is a vertebrate, you can go to Safeway and buy a bunch of eggs and do experiments on those. Okay, so those aren't live vertebrate animals, but a live vertebrate animal is, for example, a salmon embryo, right? Because it's going to be a vertebrate when it grows up. It might not have a backbone now, it might not have a spinal cord yet because it's just a zygote, but it is a vertebrate. Okay. Students can't be an investigator. That's on there because of the class that we had when we did the lab component, just so you know you couldn't do that. And then all these protocols have to be reviewed by the IACUC and approved before you can do anything, as I said before. Okay, so what the IACUC does, and I've been on it for a number of years, Dr. Krubitzer has been on it for even longer, so we're very well familiar with what goes on in these meetings. Okay, and just out of curiosity, how many protocols do you think there are for live vertebrate work at UC Davis? I mean,
They're still being created.
Pardon me, how many?
There's still more being created.
There's generally a steady state, right? So as new ones come in, old ones fall off because the experiments get done, right? So just to give you a hint, I think Harvard has on the order of maybe like 600 or something. Do you know how many we have, Leah? The exact number?
No, I don't, but it's got to be
It's like 12,000 or something.
It's got to be over 10,000. Yeah.
Yeah, it's something like 12,000. We're a land grant university. We have tons of biologists. We have all, people are doing things with sheep and with goats and with cattle and
The
with, go ahead.
the only institute that does more animal research than UC Davis is the NIH. We are the second largest animal research institution in the country.
Okay, so there you go, right? Little old UC Davis is number 2, second only to the NIH. And that means that there's an awful lot of work for the IACUC to do, right? So they review all these protocols every 2 weeks. They spend a couple hours going over these things, and it's made up of dozens of folks. And what they're looking for is to make sure that you're following the government principles and that you have considered, the 3 R's, the 4 R's, reduction, refinement, replacement, and so these are the 3 R's, and the 4th one is responsibility. Okay, so the key to all this is that it's a privilege to do animal research, and you have to treat the animals under your control humanely and with respect and be responsible about them. And the IACUC takes that very, very seriously. Okay, so what do you have to do? You can't just say, hey, by the way, I have this great idea to cure world hunger and I need 10,000 mice. Is that all right? Right? That's not going to fly. What you have to do is say the species that you're going to use, how many, and why you need that many. You can't just say I need 10,000 because that's what I've done in the past. That never flies. You have to do a power analysis. You have to say these are experiments I'm going to do. I need 8 mice per group. I have 100 groups, so I need 800 mice. That sort of thing. You have to say where you're going to do this. That way, as I'll talk about in a second, when people come to inspect you, they'll pull a protocol out and they'll look and say, Dr. Rickenson, where's he at? I want to see his operation. And it says right there, and they get in the car and they come over and look at my lab. And you have to have a veterinarian and you have to say where you're getting the money.
We have one question real quick, which is how do you metric responsibility?
That's a great question. And there's not a simple rubric, right? So it's by, and we'll talk about this in a little bit, it's what a reasonable person would say is appropriate. And we'll talk about that in a bit. Okay. You have to describe the objectives and significance of the study. Significance is important, which we'll talk about in a few minutes too. Right? You have to show that you're doing something that is appropriate to do in animals and that there's not a better way. Okay, so you can't say, well, I've been doing this sort of experiment since the 1970s. It's like, dude, you can do it way better now if you just use a different technique, or unnecessary duplications. Like, it's already been done. There's no reason to do exactly the same thing again, just to say that, it's true. Right? So you got to make sure that it's unique and the most efficient and appropriate way of doing these experiments. And then you describe what the procedures are and the drugs, if you're going to use any drugs. And the veterinarians are going to be looking at the drugs to make sure that you're not using ether instead of, some other anesthetic, etc. You have to describe what could go wrong that you expect to have happen. Right? And not every experiment goes perfectly. If you're going to do a surgery on a live rat, sometimes, something happens that you can expect them to, for example, maybe have a headache, right? After you do brain surgery on them. And then you can say, and so I'm going to give them an analgesic, right? You can mitigate the potential adverse effects that way. You have to describe how you're going to euthanize the animal if you're going to. You got to say who's on there and what hazardous materials you have. And the staff roster is important because everybody who's on that, A, has to have read the protocol and know what's going on, and B, be appropriately trained. So they're looking for training at that point to make sure that everybody who's going to be doing these procedures has adequate training for those procedures.
Does that mean everybody who is part of your lab or everybody who's taking part in the dissection? Or
everybody who's taking part in the experiments of that protocol. So for example, in my laboratory, I have people that just do computer programming, etc., and they're not listed, right? Because they never go back in the lab and they never touch the animals, right? But if you're going to be back in the lab and going to be doing some part of that, then you have to be listed and know all of it. Okay? We good? Okay, so let's touch on a couple things. So reduction in animals. So the idea is to use as few animals as possible. Species justification. You want to make sure that the species that you're using is the appropriate one. And we'll also talk about the overarching philosophy of using animals in research in general. Okay, so reduction of animals. In the 1970s, a group of perfectly reasonable scientists used 20 monkeys in a set of experiments to understand the responses of neurons in the auditory cortex. This was a seminal study. It kind of described to us how the auditory cortex is organized in the primate. Then 10 years later or so, 5 monkeys were used in a similar set of experiments. They replicated those experiments from the 70s and extended them to 7 new aspects of how the brain is functioning. Okay, so they've got essentially 8 answers for 1/4 of the animals, just 10 years later. Oops. And then 10 years after that or so, all of those findings were replicated in 2 monkeys and extended even further. So there's one issue of unnecessary replication. So all of these studies did, these 2 studies replicated findings from this one. It was not unnecessary because you needed to do that in order to extend them to make sure that everything is okay. Okay, so you're adding on. It's not that somebody did an experiment in 20 monkeys and someone did the exact same experiment in 5 and then later in 2 and got the exact same result. That's not okay. So what's the difference? Attitude and technology. Okay, so what would happen in the 70s, which was perfectly reasonable at that time, is you would get your animal and anesthetize it and do your experiment until, you know, dinner time, maybe even later. And then, you know, time to knock off, euthanize the animal and go home. Later, it was realized that, you know, what we probably should do is, since the animal's anesthetized and everything we know about this level of deep anesthesia, like we do in human patients all the time, is they don't remember anything about it. And so why not, instead of us all just going home, we'll get more people to come in and work overnight. And so we'll do 24 hours instead of just 10 or 12. And in fact, if we're going to do 24, the guys who did the experiment in the beginning are all rested so they can do the next day, and let's just drag it out as long as we can. Not drag it out, but extend it out as long as we can. Get as much data as we possibly can from each individual animal. So suddenly you can get 4 times as much data from the same animal if you just work longer. Okay, so that was the difference between the 70s and the 80s. And then later it was realized, well, if we don't actually have to anesthetize the animal, if we can do it while they're awake, we can do an experiment every day. And then in only 2 monkeys, we can do, you know, 4 times, 5 times more data collection in a single animal than we had done before. Okay, so now we've been able to drop that number down to 2. Okay, so how do you do that? And that's the technology. So back in the 70s, boy, if you had a 400K floppy drive, you had lots of data there, right? There's more, you know, computational horsepower in my little Fitbit here than there was in the computers of the day back in the 70s, right? So you would draw things on butcher paper, you would use a stopwatch to time things, that kind of stuff, and write it down in the book, right? As computer technology is advanced, you're able to do a lot more, and you can store the data for a lot longer, and then you can have your computer guys later analyze the data from even 10 or 15 or 20 years ago and find new nuggets of knowledge in those. Okay, so the technology has really helped a lot too. Okay. Okay, and I just want to point out the attitude part. Back in the 70s, it was perfectly reasonable to do the experiments the way that they did them, right? Nobody thought differently about that. This came up, how do you justify responsibility? And this is kind of an answer to that question, right? So the norm of the way to do things in those days was X, and that seemed perfectly reasonable to everybody. Now we look back and go, what a waste. IACUC would never approve that. They would never say, oh, you're only going to use this animal for 8 hours and then euthanize it. Why don't you keep collecting data from it? Right? Now we know that you can do that, so our attitude has changed over time, and that's what we expect. And I often wonder, and I wrote this in the lecture notes, so what are we doing now that's, you guys, when you're in my age, are going to think, God, I can't believe we used to do that. That was just lame, right? Instead, now all we do is blah, blah, blah. Okay, It's a good time to pause for questions if there are any.
We are good if anyone else has any questions.
Particularly. I think, yeah. So you said like, okay, that you would use less monkeys or the same amount of monkeys, but you have to extend the reaches or the understanding based on those monkeys.
Yeah.
To justify the use of them. So I guess my question is, it's only for extension of understanding or observances. It can never be for investigation of a underlying principle or of a what's the word? A generally accepted rule.
Like a dogma.
Yes, sorry.
Okay. Yeah. Okay. So let me try to clarify that a little bit. So here's one scenario. 1972, Bliss and Lomo described LTP in the hippocampus of a rabbit. Since then, there have been thousands, if not tens of thousands, of experiments on LTP. Okay, so you might say, well, that's useless. We've known for 50 years or whatever that LTP exists, but now we know the different molecular mechanisms behind it, how the different neurons are doing these other different kinds of things with very sophisticated methods, and how that relates to human memory and all these other kinds of things. So on the one hand, it's been replicated a bunch. On the other hand, now we understand all the way down to the molecular mechanisms, which channel and how long the channel is open, and what G proteins are activated and what cascades are involved and all these kinds of things. And we still are scratching our head about, well, how does that synapse know to change and not the one next to it, right? So they're still doing experiments on LTP to this day. Okay. So in that sense, we're extending knowledge and just, as I think you intimated, as to better understand the basic concept of what's going on underneath it and to challenge dogma if that is necessary. Right?
Can I for a second? We're gonna talk a little bit about this, about general principles. And so one thing that's really important to understand is almost everything we know about the human brain comes from work on other animals, because there's this thing called evolution and homology, and there are general principles of organization and function that all species share due to common ancestry. So most of what we know about the action potential doesn't even come from work on a vertebrate. It comes from work on a giant axon of a squid. So sodium and potassium channels haven't changed very much. And I see another question here. Are we going to learn about specifics of species justification? And we will talk about that. I think it's going to be sort of strewn throughout our entire lecture. For example, DARPA funds a lot of bee research. They also fund a lot of bat research. It depends on what your question is. So there's clinical relevance. You might want to work on macaque monkeys if you're interested in bimanual coordination, because they have hands with opposable thumbs that look very much like humans, and their brain areas that are associated with use that look a lot like humans. You don't want to do that on a rat because they don't have thumbs. Their thumbs are retracted. So there are it depends on what your question is and about what species you would use. And, a lot of funding agencies only want to fund translational research, but you need to know about general principles as well. So Garrick, I think your name is, did that help answer your question? Okay, cool.
And then just one last one where someone asked, instead of having multiple animals in one experiment, you use one. Do you just start doing multiple separate studies on just the same animal?
Right, that's a good question too. And so one of the things that's not on the slide is, where did you get the animals and what are you going to do with them afterwards, right? So on the one hand, there are a number of experiments where you can use the same animal and do multiple different experiments on that one animal. And for example, if you just want to observe its natural behavior in large enclosures or smaller enclosures or whatever, and you don't really have much in the way of adverse effects on that animal, commonly you'd be able to use the same animal for multiple experiments, much like I was a psych minor when I was in college, and I was a participant in many psychology experiments just because you had to get the grade, right? So that's perfectly benign and you can do that. On the other hand, what they don't want to have happen is multiple survivor surgeries or multiple manipulations that have adverse effects. So you don't want to have an animal that ends up being run through the ringer. So it's really important to know that what the adverse effects are in the history of the animal and what you're going to do and what you're going to do with it later. Okay, so the answer to that question is kind of, I guess.
And then one last question. Did you say that they use anesthesia to make them forget?
Not to make them forget, but we know that, okay. So in those
You know how
experiments,
anesthesia works.
pardon me?
Maybe we don't know how anesthesia works. It's just to make sure you don't feel it or it paralyzes you. Depends on the person.
Right, so that's a very good point. So we're studying the brain, right? And so on the one hand, if you anesthetize an animal, we know that anesthesia does what? It affects the brain, right? Because you lose your memory, you lose your consciousness. So your neurons probably aren't doing the same thing when you're anesthetized as when you're awake versus even when you're asleep versus even when you're alert or bored, right? So your brain activity is going to be different under those states. And as a scientist, we want to understand how the natural human brain works, right? And we're using, as Dr. Kribitzer said, we're using animals to try to infer how that must be. So using anesthesia is a problem to do that in that sense. It depends on where you are along the pathway where anesthesia has the most effects. Okay, so back in the 70s, it was reasoned that A, you couldn't do these experiments on awake animals, and B, that we're early in the brain pathways, so the effects of anesthesia are the smallest, so we use anesthesia to do those experiments. Okay. Those kinds of experiments aren't done anymore for those reasons. They're done in awake animals now. And they're done in awake animals that are actually listening to sounds as opposed to sleeping or just being bored. Okay, so that's an evolution of the technology and the attitude that we're not going to necessarily use anesthesia if we don't have to, because it's going to screw with our results. Okay, does that make sense? This is a real lively discussion. I'm really appreciating this.
Yeah. Can I just add to that? And a lot of scientists still use anesthesia, and we do. And the main reason is to prevent pain. But some anesthetics also are hypnotics, and so there is an element where you forget. But anesthesia, as Greg said, it changes how the brain works, but there are a lot of basic principles you can investigate using anesthesia. And in terms, I think I've worked on over 45 different mammals, and the hardest thing is to figure out the anesthesia. Different animals respond differently to the same anesthetic. Different brain regions you can study really well under a certain type of anesthetic and not so well under a different kind of anesthetic. So anesthesia is a really tough nut. And if you look at IARC protocols, there's a long expanded section on anesthesia, what you're going to use, what dose, when you're going to use it, why are you using this sort of anesthesia instead of a different kind of anesthesia. So we sometimes have 5 different types of anesthesia, depending on the animal, depending on the brain area. So it's a tough one, but the major goal is you don't want the animal to experience pain. And I know that when I'm doing brain surgery and recording from neurons, an animal is not experiencing pain. And afterwards, if an animal ends up recovering, for example, you use an analgesic, which is when the animal's conscious, you're cutting down on the pain and the discomfort. So it's a really important thing, not just for Ayakuk, but as an individual human being on the planet Earth who loves animals, is interested in research, but certainly doesn't want your animals to suffer. These are really big, weighty questions that we don't just think about every now and then when we're filling out our animal protocol, but we think about all the time.
Right. Okay. Okay, so let's talk about species justification just for a minute. Dr. Kribitzer touched on that a little bit. Back in the early 1900s, Sir Charles Sherrington, who's like the godfather of all electrophysiology studies, he studied monkeys and great apes. He was a neurologist who was interested in strokes, and he reasoned back in the late 1800s and early 1900s that we should probably use chimps and gorillas because they're the closest to humans. And it would make sense to do experiments on them. Okay, and we don't use those animals for that anymore, obviously, right? So what you have to do, is think about what's your question and what animal can you use to answer. So if you are interested in mitochondrial enzymatic processes, which a lot of people are, even on this campus and elsewhere, you can get away with yeast, right? They're a eukaryote. Our mitochondria are very similar to theirs. You can answer the same basic questions. As Dr. Krubitzer said, if you're looking at action potentials, you can use invertebrates, right? Why not use a squid instead? You don't need to use a monkey for that. Central pattern generators, sensory transduction, invertebrates, fish, and amphibians. Now, when you're doing the invertebrates in the yeast, you don't need IACUC protocols. Okay. When you get to fish, fish is a vertebrate and amphibians, and now IACUC gets involved. If you want to look at limb movement, you're obviously not going to use a snake, right? So you can use other reptiles. Rodents are not uncommon. Carnivores, depending on what kind of limb movement you want. If you want to do bimanual coordination, then you should use a primate because they do that. Sensory motor transformations or perception is commonly done in different kinds of monkeys. If you want to do emotional context, language, and things like that, you can't use a rat model very effectively always, right? Sometimes you have to go to humans. Right. So there are certain processes. If you want to talk about the most elemental parts of something more complicated, you can use a non-primate species. But as you get to more and more sophisticated human-like perceptions and behaviors and thoughts and emotion, which are very hard to define, then you're going to have to go to humans. And we'll explain how you can do that as well.
Could we also perhaps use dolphins or pigs for emotional context and language? They have been shown to understand emotion.
Right. For example, if you are interested in that, you might want to use a cetacean, but there are complications with that as well, right? So we'll talk about that a little bit, I think, in a minute. Or you can use echolocating bats, right, to see if you can understand how auditory processes work that might subserve things like language. But human language is tricky, and there's lots of linguists out there that define it such that only humans can have it, in which case, you know, you got to think about it. Okay, so could you use dolphins, etc.? So say you've done the math, you know how many animals you need or how few you need to answer the question you have. You've considered which species will be necessary. You have strong arguments why the chosen species is appropriate. And now the question is it worth it? Is the answer you're going to get worth the effort that you're making and the number of animals that are going to be used in the process? So you can think of a couple of different scenarios, right? And so I made some up. So imagine that there's hemorrhagic fever. Imagine a new kind of hemorrhagic fever occurs, and hemorrhagic fever is bad news, right? So your blood vessels burst inside of you and you die a gruesome death. Right? And imagine that one comes along and you realize that it infects humans, and it infects human children, and it kills virtually all human children by the time that they're 5 years old. And now you're going to think, well, we should probably cure this, because if we don't, humanity will become extinct, because if nobody can be older than 5 years old, then people like me will just go ahead and die, and there's going to be nobody to replace me. So should you use mice versus kittens versus pygmy chimpanzees, and how many should you use, right, to cure this disease? Is it okay to use 10,000 mice? Is it okay to use 10,000 pygmy chimps, right? Is it worth it? Well, on the one hand, you might say that it's saving all of humanity, so we can use all of them, right? And others might think, you know what? Humans have had a good run. We're kind of jerks to the planet anyway. Maybe it's time for us to say goodbye. COVID-19, this is something relatively new. Same question. Can you cure it with mice versus kittens versus pygmy chimps? And how many are you willing to sacrifice in order to find that cure? Okay. So these are philosophical questions. And so what I thought I would do is try this. I have never done breakout rooms before. There's 65 of us, so I'm going to make, I don't know, how about 10 breakout rooms. You're going to be randomly assigned. We'll give it about, what time is it? 55. We'll give it about 5 minutes, right? This is just kind of a cleanse the palate then, because I'm going to start talking about something a little bit different after this, right? So I just want you guys to meet each other and say hello to each other, and just, talk about these kinds of things, right? I mean, what do you think is the most appropriate way to do this, et cetera? So I'm going to create, I think I said 13. And we'll go 5 minutes, so we'll stop at 10 o'clock, okay? And and good luck. I don't know if I can sneak in on you or not, but here we go. Open all rooms. this. Okay, how was that? Okay. Okay. So like I said before, I'm looking for feedback. So if you want to send me an email and go, dude, you totally lamed out on the breakout room, do it this way instead. I'm totally open to that. Okay, I think most of you are back. So now let's look at some brains. But before we do that, wait, I had one more point to make. So hopefully you had some lively discussion and you got to hear some other kind of different perspectives. So what is justified is not a simple answer. So opinions are going to vary across context and individual. And I consider this idea of a continuum. So on one end, you could never harm any living thing for any reason. Okay, that's incompatible with human life. You got to eat a plant or something, right? Otherwise, all you're eating is salt and you'll die. Or you can do whatever you feel like to any living thing. That's not going to work either. You can't go around shooting people because they snap their gum or something like that. That's complete chaos and it's not going to work, right? So different things fall along this continuum in different locations. And just to give you an idea, so people that are vegan tend to be more towards the side, vegetarian a little more to the right. There's people that are okay with fishing, right? People that are okay with hunting, and maybe it's okay to hunt birds, but not, rhinos or something like that. So you'll find different people along this continuum, depending on what it is that you're hunting. Whaling, many people in the United States are against that. And you think, okay, well, whaling is way over here towards the right. It's probably a bad idea. We should try to stay towards the middle. Whenever I think about this, I think of a group of Native Americans that live up in Bering Sea, and they have this ceremony every year where they get in their kayaks that they make out of whatever is up there that you can make things like this out of, and they have their pointy spears and they go try to kill a whale. And as far as I know, they've been unsuccessful at this for multiple generations, but it's part of their religious ceremonies. And on that one hand, if you're going to paddle a kayak with a pointy stick and try to kill a whale, which you almost certainly aren't going to succeed at, for a cultural benefit, that puts it more okay compared to, well, I'm going to use some high-powered machine gun thingy to kill a whale so I make money, right? So even a concept of whaling can fall along this continuum from being, more or less okay to some people to absolutely wrong under any circumstance. Okay, it just depends on who you are. Okay. So given that, let's talk about brains for the next 15 minutes or so. Make sure I don't go over. Okay, so this is a human brain. Okay, this is if you take a brain out of a skull. This is of a person. This is the left side of their brain. It's as though they're looking towards the left. Okay, and over the course of the next few lectures, we're going to come up with a terminology so we can talk about this structure in a way that isn't the way I'm going to talk about it now. Okay, so there's these, if you look at this, and you're uninitiated, you might think this is like small intestines. It's a bunch of tubes, okay? But it's not. In fact, it's one entire sheet that gets crumpled up so that it fits inside the head. Okay, and you can see that if you make a cut along one dimension, like down here, you can ask, well, what's going on inside these places where I can't see? And first, here's a human brain, and then a rhesus monkey brain and an owl monkey brain. So these are 3 different species of primates. These are, through the magic of PowerPoint, made about the same size. This is to scale, right? So you can see the human brain is way bigger, right? And it also has more of these tubey things, right? So there's more crevices or dips in here compared to the rhesus monkey, compared to the owl monkey. So I think, yeah, now let's take a cut. And what you find is this part right here is the same, is right here. Okay. So if you ask what's going on inside this, crevasse here, the answer is this: it goes in and there's just lots more brain, right? So what you can't see in this one little space here has more tissue, that's neurons, than the whole of the owl monkey brain. So you can see that there's an awful lot of gray matter, which is blue in this case, in the human brain compared to the rhesus monkey brain and the owl monkey brain. And we're going to talk about these kinds of things quite a bit, how evolutionarily things have happened such that you end up with more cerebral cortex in some species compared to others. Okay. So back in the 60s and 70s, a handful of folks, Clinton Woolsey, Jersey Rose, and Wally Walker, embarked upon a series of experiments along with John Koss, shown here in his youthful exuberance, to ask, what do different brains look like across different species? And what can we learn from that? Okay, so here are kind of the classic experimental models of those days: the cat, the dog, this is a beagle, and the mouse and the rat that are shown at about the same size, so they're scaled differently. Okay, and you can see that there are some similarities and differences. Similarities include there's this part back here, which is called the cerebellum. There's this part up front, which is called the cerebral cortex. And there's this part at the very front, which is the olfactory system. And then there's this part back here, which is the spinal cord. Okay, and then as they had access to a number of different zoos, they could look at other species. So here's a more exotic species like kangaroos and polar bears and armadillos and Tasmanian devils. And you can, as you walk through this website, which I like to do, and I consider it, you know, like preparing for class, but it's actually quite a bit of fun. You can begin to appreciate the different kinds of structures that are seen across different lineages. So for example, if you look at animal like a porpoise, this has an awful lot of gray matter here, right? There's an awful lot of tissue that's devoted to the cerebral cortex compared to, say, manatee, which only has one of these little things. Right? Here's a duck-billed platypus. And what you're seeing here is this view is sort of like the one that I showed you, the human. It's the left side. This is the right side. This is kind of towards the top. This is underneath, and that's the very top. So you get an appreciation of different ones. You might ask, well, why is this kind of all weird? What are these blood vessels and stuff there? And that's due to the way the tissue was prepared. Okay, so it depends on what you're going to learn about in the next couple of weeks, is how do you make this tissue look so that you can see things better and not messed up by the blood, etc. Here's an echidna, which is kind of a cool little critter. Here are places where electrodes were used to define what the receptive fields were like in this little guy. Here's a dog. We talked about that before. And what I want to do is show you a bunch of different dog-like creatures. So if we look here, say at the left side, you see that here's the cerebellum and the spinal cord and the olfactory bulb. And here's this structure that looks kind of horseshoe-shaped. Here it is in a wolf. You see the similar kind of horseshoe shape, or a coyote, or a red fox. It's real prominent here. And even a cat, which is a carnivore too, right? It has this kind of concentric horseshoe-like configuration of its cerebral cortex. Here's a leopard, similar organization. African lion, shown here. And then you get to a pig. Pig's not a carnivore. Pig's completely different than that. And its brain structure looks a little bit different too. In fact, if you were to be handed one of these and one of these, say, and asked which one is the carnivore, you should easily be able to say, oh, this one's definitely the carnivore over this one. Where is it? Because it doesn't have that. There's this thing weird in the middle, right? It looks like it has two. It looks very different than that. And pigs have quite a bit of cortical horsepower. So like to think of that as a reflection of their, their cerebral sophistication, intelligence, in quotations. Here's a goat. Goats are apparently pretty smart too. But again, it looks more like a pig than a carnivore, right? Because it's got this thing in the middle. And here's a sheep. You're going to see a lot of sheep brains because this is what Dr. Krubitzer cut up in her kitchen with her mother. There's a white-tailed deer. I never realized that white-tailed deer had so much cortical horsepower as they do. When I was a kid, my dad was a deer hunter and he would take me hunting. I should have known that they were actually really smart because we never saw one. They always ran away from us and we never even got off a shot. So they're smarter than my dad anyway. Here's a llama, which is apparently even smarter than that. Camel, this is the dromedary camel or the one-humped camel. Okay, here's a lesser horseshoe bat. So this is a small animal in the first place, but it's got a very smooth brain. It's called mesencephalic, which you'll learn about. And here's the top and the bottom, and we have better words for that I'll let you learn in the next couple of lectures. Let's look at primates now. So here's Here's a flying fox. Now here's a primate. Okay, so primates have unique, and they have this structure here, right, which I showed you before in a human, right? And then we're going to see in this structure here. So it's divided up into different, what are called lobes. This is so-called temporal lobe, occipital, and parietal, and the frontal lobe. And you can see the owl monkey. This is a nocturnal monkey, lives in South America, has a tail, so it's called a New World monkey. Here's a squirrel monkey, very similar. It's got a very similar kind of brain, maybe a little bit more of these, what are called sulci. Okay, marmoset, much different, smaller, smaller sulcus here. Howler monkey is starting to get a little more complex. TD monkey, they look maybe a little bit deeper, so there's probably more cerebral cortex there. Here's a spider monkey. You're beginning to see the emergence of other of these sulci. Here's the rhesus monkey. This is probably one of the more studied animals, primates, for neuroscience research. Here's a langur, and then a baboon. And you can see that all of these primates, this is very different than the carnivores, right? So the carnivores had those horseshoe things, and the pig had a thing in the middle, and these guys have the really prominent temporal lobe with various number of sulci within there. This is a gibbon. Now we're at the apes. Here's a gorilla, which is hard to see anything. I guess here you can see the prominent temporal lobe here. This is organized a little bit differently than the other ones. And then a chimpanzee looks very much like a human brain, I would say. It looks very complex. And then here's the human brain, which is even more so. I hope you all know who this is. This is our leader. This is Chancellor May. I've had, well, I'll skip that for now. One of the things that becomes apparent, and you'll learn a lot about in this class, is that if you look at primary cortical areas, and you're going to learn more about these, but this, so for example, the primary auditory cortex, that's the part of the cerebral cortex that gets the first input from the lower nervous system about audition. Primary visual gets the first information in the cortex about visual information, and somatosensory is green. And what you notice is that in all of these species, the vision is towards the back, which is what's called the occipital pole. The auditory in blue here is on the temporal lobe, and then the somatosensory is right next to the motor cortex, which is right here in the parietal and frontal lobes. And you'll probably see this slide a lot too. If you look at the visual, auditory, and somatosensory cortex across all these different lineages, what you find is that they're very consistent in that the farthest back part, or the farthest caudal part, is visual cortex, and then the motor cortex is towards the frontal end, and the auditory in green in this case is towards the temporal side. Okay, I think that's it for me. Okay, are there any questions about anything up to this point?
Yeah, if you go back to the slide.
See if I can.
Okay, maybe just an evolution question then. You said earlier that there's homology between all vertebrate brains as far as their structure and function. However, I was also wondering if that extends to the amount of SUSI or gyri. There's a previous myth that the smarter a person is, the more wrinkly their brain is. And I was wondering if there is some, not a contradiction, the confoundation, when things kind of line up to look like that.
So the myth is, you're saying that the more crinkly the brain is, the smarter you are.
Yeah, is that kind of true across animals, not just in a specific species, but if you compare a sheep brain to a mouse brain, which I believe the sheep is smarter, or a crow brain to a mouse brain, would the crow brain usually be wrinklier?
Okay, so there's a couple of layers to that. And let me I'll let Leah jump in here if she feels like it. But the basic idea is that the more cortical sheet you have, the smarter you are relative to your body weight, right? So for example, if you plot the cortical sheet size as a function of body weight, it usually forms a nice, is it this way? Nice linear function, okay? With the exception that primates are up above it and higher. So for the same amount of body weight, they have more cortical mass. So they're smarter, right? And cetaceans actually are above that even, but we don't talk about that because that makes us look bad, okay? So the bigger you are, the bigger your brain's gonna be. And that makes sense because you have more, you know, body surface to encode, et cetera, right? But also the bigger your brain is relative to your body weight is linearly related to intellect, if you want to call it that. Leo, you want to put me into that hole?
Right. So the cortex can expand in two different ways. One through concerted evolution, in a way that Greg talked about, where you have changes in developmental programs that affect the whole body and they affect the brain, and the cortex gets larger, and it will get larger than the rest of the brain. And then there's mosaic evolution, where very specific parts of the brain can get enlarged independent of other parts of the brain. And the neocortex in primates has done that. So it's not about whether it's crinkly or not crinkly. It's about the size of the cortical sheet overall, but also the relative size of the cortical sheet. And you have to be careful when you're comparing mammals with birds, because there are crows that use tools in the wild and are smart. And I don't like to use the term intelligence or smart because it's very anthropocentric. The biggest stumbling block to understanding the brain or the human brain is the nervous system through which we study it, which is our own. I prefer, and throughout this course, I like the term complexity, because intelligence is it's a human term, and trying to apply it to other animals, I think it makes it really hard to understand brain evolution in general if we talk about things like intelligence. And so, you know, platypus is super intelligent given the context in which it developed, in which it lives. And all species are super intelligent based on the context in which they develop and they live. And I think through Greg's lectures, you're gonna, you know, this will become more apparent, and hopefully we'll stop using things like intelligent, and maybe complex is a better word. Behaviors that have many parts versus behaviors that are very simple. Okay, I guess that's what I'm gonna say about that.
No, that's good. I'd say intelligence, but I have to use air quotes because.
Okay, so we have two questions. So the first one is pertaining to this lecture, and it says that the lecture that's posted on campus with pre is not the same, does not have the same slides that the information you just presented. And will that be updated with the information you just presented?
Yes. So what I do is I have the pre one, which is what is good for you to look at before you come to class, right, and read the notes. And then actually I'm going to post this Zoom call, right? So you'll have all the slides and me talking over them. So, and that will be in the post folder on the Canvas site.
Okay, cool. And then you talk about the cortical sheet size, but is relative size to of the matter wrapped inside the cortical sheet a factor to its complexity?
Can you explain? I'm not certain what you mean by that question.
I guess you talk about like you say like the cortical sheet. So is that just like the matter that's wrapped? Oh God, sorry.
Right. Oh, right. Okay. So the cortex means bark. It's the outer part. And we're going to have a gross neuroanatomy lecture on Wednesday. So you'll understand what the cortex is versus hopefully the thalamus and other portions of the brain. But I think a really nice example is if you look at a mouse lemur and you look at a rat, they both have the same, and a mouse lemur is a primate, and they have complex behavior. They both have the same size brain. It weighs almost the same amount. I think it's like 3 grams or something like that. Maybe it's a little more. But the amount of cortex that a mouse lemur has compared to a rat is about 2 or 3 times the size of a rat brain. And the cortex is the stuff that's involved in cognition, perception. In humans, it's involved in language. It's that part of the brain that's involved in this complex behavior. Whereas in the brainstem, there are nuclei that regulate your heart rate, they regulate the diaphragm for living and breathing, for just basic life functions. The spinal cord is about motor circuits and activating muscles in the body. Whereas the stuff that we identify, that makes us human compared to, let's say, our dog, is the neocortex. And you guys will learn a lot more about that because both Greg and I are kind of neocortex people. So does that answer your question?
Yeah.
Cool.
Okay, so we're technically out of time now, but also both Dr. Krubitzer and I have our office hours right now. So if you got something to do, you've had it. Thanks for your attention. We'll see you on Wednesday. If you have any more questions, we're here to answer.
I have a laboratory question for both of you. Will there be any open positions within the fall or over the summer within your laboratories?
Well, that's a very nice compliment. We didn't scare you off after the first lecture. Oh my God. You know what?
This is brain. I am brain. I must learn brain.
Yeah, I'll tell you what, under normal conditions, most of the undergraduates who work in my laboratory come from classes I teach. And I think if things go well in the fall, sure. We have a lot of undergraduates who work in our laboratory who, depending on the amount of time that they have to commit, will be involved in a variety of different things: microscopic analysis, cutting brains, histological processing, analyzing behavioral video. Greg, you want to say something about this?
Yeah, I do the same. So a lot of my undergraduates come from this class too. They do data analysis mostly and things like that. The last two lectures are about what we do in our laboratories. So those will be, if you're still inclined at that point, you'll have a better idea what we do. The caveat, of course, is we're in COVID-19, right? So undergrads in the lab are something that's frowned on right now, and it's all remote, and it's kind of a weird thing. So hopefully, whether you're interested in coming to my lab or not, I'll be able to have undergrads in my lab this summer, or it's just weird right now. So
I understand, but you're my professor. I ask the same question to all my professors. Hey, do you have an open position? And I'll try to do better dissertations later. And thank you for informing me.
Yeah. Greg, do you want to stop the recording?
There is one question. This from Josephine.
No. Okay.
Are office hours recorded?
yeah, that's fine.
Oh, they are?
I haven't stopped recording yet.
Oh, the office hours, like in general, are they recorded?
Well, so my office hours are right after lecture, so I don't even know how to stop recording. So that answer is yes. I don't know, Corey, what you're going to do with yours. So that's your question.
Yeah. I mean, I for now probably not, because I feel like depending on how many people show up to my office hours, it might be a whole hour of nothing, So for now, I'll probably leave it off. But if it's consistently wanted and people want my office hours recorded, I'd be happy to do so. But as of right now, probably not.
Okay. Thank you. And Dr. Kruzabit, sorry. How do you say your last name, Dr. Leah?
Oh, Krubitzer.
Krubitzer. Sorry about the emails I sent this morning during class. I couldn't find the Zoom link.
Oh, no worries.
Yeah.
Well, you made it, so that's the good news. I got on early. I was scared to death. I was the only one until like 56 or so. I was like, oh my God, I totally screwed up. Everybody's sitting in some room somewhere.
Greg, I did that last quarter. It was my first time teaching on Zoom, but I only had a small class, and I was there really early. I was so nervous that I was going to screw things up.
Oh, sorry.
So, when would the TA's office hours?
My office hours are on Tuesdays and Thursdays from 6. It's always hard for me to rethink it because I'm based on the East Coast. So it's 3 hours behind when I have them. So they're from Tuesdays and Thursdays from 6 to 7.
Okay, so 5 to 6 is...
Wait. 8 to 9. No, sorry. It is from 5 o'clock to 6 o'clock.
Okay, so those are yours. Okay.
Yeah, those are mine.
Okay. Thank you.
Yeah, sorry. I'm always having a trouble with the time shift since I've been based here for so long.
So you said you were gonna post the recording, but I guess I was just wondering if you were gonna post just the slides, like without the recording, so I wouldn't have to go through the recording again.
Sure, I can do that.
Okay, thank you.
Okay, so I'll make a folder of lectures post and lectures recorded.
So for your office hours, would it be the same Zoom that we're on, or would you be sending us different links every week?
Same Zoom. I
Same
mean,
Zoom,
we
okay.
might as well just extend the same Zoom, because the
You're already
reason
here.
I have office hours after class is because that's when people really have the most questions, and you know, the material is really fresh. And so obviously you can email me or Greg and, you know, ask questions, and if you really need something explained in detail that we can't do after class, but generally it's just be part of an extension of the lecture.
Okay, thank you.
Yeah, so I scheduled these lectures from 9 to 11, right? So we have until 11 o'clock, as long as you guys want to stick around. And commonly when it's live, that's what I do. And I get a handful of students to come up almost after every lecture. And you know, today you can only ask about lecture 1, but you know, in the future, you can ask about this lecture again too, if when you're studying something comes up and you don't understand it. So that's totally legal.
So I had a question out of interest. If nobody has any logistical questions or anything, go ahead.
I just typed in the chat. Do you think you could post the Zoom link on the homepage just so it's always there instead of the announcements? Because then you have to look for it.
Yeah, it is. It's on the first page. I think it's a PDF.
Oh.
Yeah, there's the syllabus. There's the student conduct, and I think it's called Zoom invites or something like that. And it's got the whole shebang with all the phone numbers and all that stuff.
Thank you.
Yeah, you should be good. Okay, I think, Inara, did you have a question?
Yeah, so I work at the IACUC, and I've been working there for about a year and a half. And this is the first class that anyone's ever brought it up. And I was wondering, as PIs, like what your experience is with submitting your protocols and all the amendments, and then preparing for the lab inspections. And just because I've been behind the scenes for it for so long, that it would be really interesting to hear like your perspectives as PIs.
Well, I used to be on IACUC. I think I was on IACUC for 7 years. And
Really?
yeah, I was at the IACUC meetings and stuff like that, and I would actually be part of those inspections. Here's the deal: if you want to do animal research, you gotta follow the rules. You follow the rules because you have to, but you also follow the rules because we are really lucky with our IACUC. Our IACUC is fantastic in terms of trying to help you do your experiments and make sure you take care of the animals. There are other universities where IACUC makes it impossible to do experiments. What our IACUC understands is that we're not running a zoo or a petting show, that the animals on this campus are there to do research on, right? And so they help you in any way they can. And look, you know, I have animals in Young Hall. I have my own lab that has to be inspected. We have field pens outside that have to be inspected. And then when I teach the lab for this class, so 4 different sites that have to undergo IACUC inspections. And my experience with them is really positive. You know, writing a protocol isn't fun because you have to put in every single detail. And then any protocol may have a number of amendments. I think I have about 12 active protocols.
Yeah, I've seen like both of your names, so it's good to see like faces with names
Like
now,
with
because I
people.
usually do the filing. Yeah, because I usually do filing, so I see your last names there, or like if I'm making spreadsheets or anything. So it's really good to see. You know.
That's right. We have a lot of different protocols. So we work on a lot of different animals. And then you have to put in amendments and stuff like that. But no, it's really positive. And I know a lot of people on IACUC from the times that I worked there, but also because I have a ton of interactions with them.
Mm-hmm. Thanks for answering that.
Greg?
Yeah, I have a similar experience. I was on the IACUC, I don't know, Lee, if we overlapped or if we overlapped much, but that was before you. And yeah, it's been I hear horror stories from colleagues from other institutions where they don't know anything about monkeys or they don't know anything about, I mean, Leah would have a hard time in a lot of different institutions where they don't know anything about, ground squirrels or whatever different species are. So yeah, they're really pro science and pro, how should I put this? They're professionals, right? They understand exactly what their role is and they do it very, very well, right? And make it as painless as possible for everybody. I can't imagine that USDA loves them too, because it makes it easy for them to do their inspections, and ALAC passes us all the time. So I think they're kind of a model, especially since the second biggest one in the country, right? Number of protocols.
Yeah. Were you active during the ALAC accreditation that happened last September? Like, did you all have to do, because I remember our office was so busy. It was, I think, September of 2019 when we had our last ALAC.
I actually was at the final meeting, and I'm the faculty in charge of an animal facility, so I interacted a lot with the ALAC inspectors and stuff like that. The previous ALAC accreditation, I was actually on IACUC, and it is a ton of work.
I remember that.
Yeah, I was involved only in the sense as a normal PI that, they came to walk through our space and all that stuff, right? And, one nice thing about the ALAC inspections is facilities comes around and touches up the paint and, that kind of stuff.
On the doors. That's so true.
So are these records or these like reports and stuff on things that we do at UC Davis, are they public, or
Yep.
do you know like where I can find them if I want to look?
Well, they're all subject to Freedom of Information
right.
Act, right? So
I think
I would discourage you from doing it just out of curiosity, but if you have a reason,
I would probably have to dig then if I really wanted to. Okay.
Yeah. I mean, the easiest way to read a protocol is to join a lab, because then you have to.
Or you can pay the fee or go to the UC Davis library. There's actually a couple of publishing houses that are specifically about protocols. I would be careful though. There's protocol papers, and then there's review papers, there's experiment papers, and then there's just the in-between research papers, which can be like the development of protocol, but it's like, if you look at the Argus 2 system, which was like supposed to be giving, if you have retina pigmentosa, Argus 2 could give you back your sight. But if you try to look at the FDA publications of how it worked, a couple of publications are like 1 page long. You kind of know this pattern of like, there's not a lot of documentation, but it's there. So just be careful when you try to do your research.
Yeah. I don't think I'm not gonna dig that deep. I was just wondering.
No, it's Freedom of Information Act, and we've had some animal rights activist groups petition to look at some of our protocols, and they get them. And the thing is, when I'm writing my protocol, they can absolutely have them. I mean, and during our experiments, any member of IACUC they can come and look at any of the experiments we're doing at any time without warning if they want. And most labs I think on campus run that way. It's like, yeah, sure, you can come, you can see what we're doing. It's approved, and we take care of I'm proud of us. We take really good care of our animals, and we care about our animals.
Yeah, no, we've never had, in the Center for Neuroscience, I don't think there's ever been an issue in our buildings with, because sometimes they go, oh, I want to go see Leo Kribitzer's operation and wonder what's going on there. And it's like, well, as long as we're here, let's go look at Greg's operation, right?
I don't know if this happens to you, but if they're there during my experiments, or if they come, they're actually, because we work on a lot of different animals, right now we're working on Megacoropteran bats and a variety of other things. They're actually really interested. They're there on an official duty, and this goes with the ALAC inspectors and the USDA inspectors. But a lot of them, they want to just talk about the animals. It's like, or they want to see the animals and just, so they're professional, but they're also human beings who are interested in, motor cortex and bats. So yeah, that's always good.
Yeah,
Speaking about freedom of information and seeing the lab, I once tried to do that. Like I tried to once walk into the Center for Neuroscience. I found I needed a card and actually be part of the lab. So when you say people are able to use the Freedom of Information Act and come take a tour or check out your research, what does that mean?
No, they can have Freedom of Information Act. They can access the records and they can access the protocols. That doesn't give them free reign to walk into the laboratory. The people who come to the laboratory at any time are members of IACUC, can come to the laboratory at any time. But Joe Schmo off the street can't just say, hey, I want to go in Leah Krubitzer's lab, whether she likes it or not. It's not really how that works.
No, it's dangerous. It's a laboratory. You have to be trained. You have to know what you're doing. Right.
Yeah.
If you drive too close to the primate center, they turn on a loudspeaker and they're like, turn around.
Really?
Yeah.
Really?
I didn't know that.
They don't do that at the Raptor Center or at any of the sheep farms. I like to go jogging around the farm areas. I'm trying to pet the sheep, but I think they're scared of humans.
Okay, anything else?
I think we're all topped out.
Everybody good? All right, well, thanks for sticking around.
Thanks for sticking around. And I mean, the class, it was a big class, but there's a lot of interaction and that makes it so much easier to teach these courses. Thanks, guys.
Okay, we'll see you all Wednesday morning.
Hey, Greg, I wanted to talk to you about something. Bye, guys.
Bye.
Bye.
Josephine and Jay, do you have anything else you want to ask us about?
Let's see. I did ask, like, is it learning objectives, or should we just remember, like, the brains of all the different species?
Right, so I will post on the lectures that I do on the post. I should have done it before. So I'll put learning objectives on my slides, right? So I don't want you to don't make flashcards of the different brains and then have to know that's a kangaroo brain, that's a polar bear brain, that's a aardvark brain or anything like that. I don't want you to do that. Okay, I want you to, it'll be a little more clear as we go here, but I want you to basically understand, basic brain organization and how it is different in different species, but not to the minutia of detail that, oh, that's a red fox versus a gray fox or something like that.
Okay, yeah.
Yeah.
Thank you.
Sure.
So I'll see you on Wednesday.
Wednesday.
Yep. Yeah. Bye.
Bye.
Jada, did you have a question? Okay.
Okay.
She was, I don't think she was live.
No, I don't either. Okay, so, hey, good lecture. And they are pretty interactive. Couple of things. One is, you don't have to come to my lectures.
I think I will.
All right. I don't
I'll kind of do like you did. Let's kind of hang back and roll my eyes and,
I'll see if I'm going to go to yours or not. I'm teaching 121 this quarter as well.
Right.
And so here's the other thing too. So for office hours, my idea was I was just going to go to office hours for my lectures. Do you think I need to be there for the whole class? For all the lectures?
No.
Okay. What I'll say is after my last series, after I do the last series, the last one in the first part, I'll just tell them if they want to talk to me about anything, you could be by appointment from then on.
Okay.
Because, like I said, my quarter is it's just huge.
My guess is that when I'm lecturing that big old stretch of February or whatever, no one's going to want to talk to you.
That's what I'm thinking too, because the first exam is going to be over.
Yeah.
Okay, cool. What else? Okay, that wasn't so bad.
Thanks. We'll see.
No, I think your lecture was great. I'm talking about the students and the student response.
Yeah,
I
yeah,
mean,
no, I am too.
some of them that were really, if you look at the chat, like a ton of them at the end thanked us.
Yeah.
Sort of like, whoa, okay, cool. All right, well, I'm going to go into my office and
Where are you
prepare
now?
for lectures. Huh?
Where are you now?
I'm at home. I was going to go into my office before this class, but I didn't make it. But I'm going to head.
Windy out there.
Yeah, I know. It's great. Because my dogs bark, if I'm giving the lecture, it's just too hard to be here. So, okay, so Wednesday, it's up to you if you want to come to my lectures or not. But I'm glad I came to yours because at least I know what you presented and what you told them I was going to present. Which I wasn't planning on. No, I wasn't planning on, but it's so easy for me to include a lot of this stuff. So good. All right, cool.
Okay.
I will see you on Wednesday.
Okay, have a good day.
You too. Bye, Greg.
Bye.